And this is due to both direct effects of the treatment, so cardiotoxic effects of chemotherapy and radiation, but also indirect treatment effects, such as lifestyle changes that can happen with a cancer diagnosis, leading to deconditioning, decrease in lean body mass, and combined with non-modifiable risk factors such as age and genetics, the risk in this patient population for the 3 million breast cancer survivors in the United States is an increased risk of 30 to 40%. And I was really struck when Graham showed me this graph. This is a graph that shows the cumulative incidence of cardiac events as a percentage in the years following a breast cancer diagnosis in an NCI-sponsored trial. And this graph is stratified by cardiovascular disease risk factors. So in the blue line, you can see that women that have 0 or 1 risk factor, and by risk factors, we're talking about family history, hyperlipidemia, hypertension, diabetes, the common risk factors. The incidence of cardiac events is around 12%. People that have 2 risk factors, it's approaching 30%. And in women that had 3 risk factors, the incidence is 50%. So 1 in 2 women in this group are having a heart attack. Or so here, a cardiac event can be a heart attack or a stroke. And we were surprised to find that people hadn't really looked at what happened after the heart attack. So the increased risk of cardiovascular events in this patient population is really well established, but no one had in turn looked to see whether MI could affect cancer recurrence or progression. And we decided to look into this. And the first thing that we did was to look to see in a patient population whether there was an association between cardiovascular event and breast cancer recurrence and mortality. And sorry, this was a retrospective study of 2 prospective case cohort studies. And we did this in collaboration with Kaiser Permanente, and these are the 2 names of the 2 populations, where patients had been followed with a mean follow-up of approximately 12 years. And we looked at people that did not have a prior history of cardiovascular disease and compared those that had a cardiovascular event, which was defined as MI, stroke, heart failure, or arrhythmia, and looked at recurrence and breast cancer specific mortality. And we saw that compared to people that had not had a cardiovascular event, women that did have an MI or stroke had a 60% increased risk of cancer recurrence. And if cancer recurred, a 60% increased risk of dying from their cancer. And so this was really striking, and to me, gave us a lot of confidence going forward in trying to figure out why this association might be appearing. These are the associated Kaplan-Meier curves, and you can see here on the y-axis percent disease-free survival as a function of time, and this is the line of people that did not have the cardiovascular event versus having a cardiovascular event, so they have a shorter disease-free time. And on the right, breast cancer specific mortality, so percent survival plotted against time, and in red, the CV event. So to begin to understand how MI alters breast cancer progression, we decided to use mouse models where we would simulate a myocardial infarction by performing an LAD ligation. So this is surgical ligation of the left anterior descending coronary artery. And this is a surgical procedure. So we had the mice that had the LAD ligation or an MI, and as a control, the control mice had a sham surgery where the suture is passed underneath the LAD but not tied off. And we combined this with several different breast cancer models. So the first one that I'll show you is the EO771 orthotopic breast cancer model. And in this model, EO771 cancer cells are injected into the mammary fat pad of the mouse. And then we induce the LAD or sham surgery 3 days later. And we followed tumor growth over 20 days. And we saw this striking increase in tumor volume in the mice that had MI compared to the sham. And you can see, interestingly, that the tumors begin to be palpable at around 10 days. And that's where we start to see this divergence. So we start to see that the mice that had the MI, the tumors are growing faster. And this was the tumor weight here on the right at sacrifice at day 20 after tumor implantation. And you can see that there's been a doubling in tumor size, basically. And when we looked at the tumors, we performed Ki-67 staining to look for proliferating cells. And on the left, you can see in green the proliferating cells compared to the sham on the right. And this seemed to be happening in both CD45 negative, so tumor-derived cells, as well as CD45 positive immune type cells. We saw an increase in proliferation after MI. To validate this in an independent model, we chose a spontaneous tumor model. So this is the MMTV-PYMT model, where breast cancer tumors appear spontaneously in mice after 14 to 16 weeks of age. And so in this model, we had to wait for palpable tumor formation, and then we induced the LAD or sham surgery, and again followed tumor growth. And one of the reasons why we chose this model was the previous one that I talked about, the EO771, doesn't really metastasize, but this spontaneous model does metastasize to the lungs. And so that gave us an opportunity to look at not just tumor growth, but also the effect of MI on metastasis. And in blue, you can see the mice that had the MI compared to sham in gray. And again, we saw this doubling in tumor growth. Down here on the bottom, we looked at the expression of MMTV, the MMTV gene in the lung. We weren't able to see overt metastases in the time frame that we had picked. You'll notice that we stopped at 18 days after MI, and that's because we didn't want to have heart failure as a confounding factor. So in this time frame, we didn't see overt metastasis, but we're still able to detect basically this tag on the cancer cells. And we see an increase in metastasis to the lung. So one of the things that people might say is, well, these tumors were growing faster and were larger, so maybe that's why you have more metastasis. But this was actually a subgroup analysis where we chose mice with similar sized primary tumors and then looked at the expression of this tumor marker in the lung. And we think that we're seeing increased metastasis. And in fact, this was from the paper, this is the published data, but we've gone on to confirm this in other models. So what could be causing this increased tumor growth? And we know that the immune system is critical in regulating tumor growth and cancer progression. And when the immune system is functioning properly, cells, anti-tumorigenic cells, such as NK cells, M1 type macrophages, and T cells can restrict tumor growth through antigen presentation and secretion of type 1 cytokines, such as interferon. But tumors have developed mechanisms for immune suppression or immune evasion. And some of the cell types that can contribute to this immune suppression are regulatory T cells, here shown in purple, and myeloid-derived suppressor cells. So these myeloid-derived suppressor cells have many of the same markers as Lysine high macrophages, interestingly. And they seem to be hung up in this immature state and don't differentiate into tumor-associated macrophages. And together, these immunosuppressive cell types can inhibit things such as cytolytic granule release from the NK cells. They can block the transition to an M1 phenotype and favor an M2 phenotype. They can block antigen presentation and inhibit B and T cell responses that might restrict the tumor. So we wondered if the MI could be reprogramming the immune system. So to look at this, we did flow cytometry of the tumors at day 20, and just looking at CD45 positive cells in the tumor, so the number of immune cells in the tumor, we saw that the mice that had experienced MI had an increase in CD45 positive cells. The proportion of those cells that were CD11b positive did not change, but when we looked at the Lysine high mDSC-like cells, we saw that after MI, we had a significant increase in the Lysine high. And by contrast, we had a decrease in these GR1 negative F480 positive cells that are tumor-associated macrophages. Looking next at the T cell population in the tumors, we saw, using CD4 as a marker, a decrease in the proportion of the immune cells that were T cells. And while we didn't see significant changes in CD8 positive or CD4 positive T cell populations between the sham and MI group, we did see an increase in the CD4 positive, FOXP3 positive regulatory T cells. And this, going back to what I was telling you about the tumor immune landscape, suggests that we're having an increase in two of the main immunosuppressive cell types in the tumors, the Lysine high myeloid-derived suppressor-like cells and the regulatory T cells. We confirmed this using an independent technique. This is a nanostring tumor immune profiling assay that allows you to take whole tumor RNA, and using a variety of markers through gene expression analyses, it gives you a readout of the tumor immune profile. And so you can see that in the mice that experienced MI compared to sham, we have this increase in regulatory T cells versus tumor infiltrating lymphocytes. And down here, a decrease in CD8 versus Tregs, suggesting again that, that we're having more of an immunosuppressive environment in the tumors. And you can think about the tumor as having an immune set point. And so things that promote tolerance, such as the mDSCs and the regulatory T cells, will suppress immunity, whereas things that increase immunity and inflammation will be anti-tumorigenic, such as CD8 T cells and type 1 interferons. And so what we're seeing is this shift towards a more immunosuppressive phenotype in the tumor after MI. So how could MI alter the immune response to cancer? We know that MI has myriad systemic effects. It induces ischemic injury, but it also initiates danger and alarmin signals, such as the release of interleukin-1 beta that can have effects on hematopoiesis. Beta adrenergic stimulation also increases hematopoiesis and generates monocytosis, which could be contributing to this. We also wondered whether MI was reprogramming the innate immune response, so not just maybe affecting the numbers of hematopoietic progenitors and monocytes, but whether it could actually alter their inflammatory set point. And by that, I mean potentially imprinting an innate immune memory of this sterile inflammatory event. So there's been a lot of interest in the last 5 years in trained innate immune response and tolerant and tolerizing innate immune responses, where after exposure to a microbial ligand and reset of the immune set point, when you expose to a second stimulus, you can either have a heightened or a tolerized response. And so we wondered whether MI could potentially be doing this. So we began to look at whether there was enhanced recruitment of Ly6E high mDSC-like cells to the tumor, whether their phenotype in the tumor microenvironment were altered, and whether we saw reprogramming of hematopoietic precursors. So we know from work from Matthias Nerendorf and others that in mouse models of LAD ligation, the LAD ligation induces a monocytosis, and it seems to have two phases. There's this first inflammatory phase, which lasts 3 to 5 days, and then a second reparative phase where the tissue repair is happening in the heart. But typically this resolves by 10 days post-MI. And when we looked in our mice, there are a lot of lines on this, so let me orient you. The triangles are the mice without tumors. So sham is in gray, that's this line here, and MI is in the red triangle. And you can see that we're seeing something similar. We're seeing a monocytosis that peaks around 10 days and then comes down. But when we implanted the tumors, these EO771 tumors, now this monocytosis was sustained. So we did not see the levels of the monocytes dropping after 10 days as we did in the absence of the tumor. And this was due to an increase specifically in the Ly6E high monocytes. So in red, you can see the mice that experienced MI have higher levels of Ly6E high monocytes in their circulation compared to those that experienced the sham surgery. And we did not see a significant difference in the Ly6E low, so we focused more on the Ly6E high. So we knew that we had more monocytes in the circulation that could potentially be recruited to the tumors, and we did some monocyte transfer experiments to see whether recruitment was enhanced. And so we used the CD45.1 and CD45.2 congenic strains to be able to trace the monocytes that were adoptively transferring. And we started with CD45.2 mice. We implanted the EO771 tumors, performed the MI or sham surgery. And then at day 12, which is at the time point before we see the tumors really diverging in size, we transferred CD45.1 monocytes. And we looked 1 day later at the number of monocytes recruited to the tumors. And this is just the tumor weight. So you can see that at this time point that we picked, there's no difference in the tumor size, although eventually there will be. But we had an increase in the recruitment. And because this is a short seminar, I'm not going to show you all the RNA-seq that we did on the tumors and the monocytes at this time point, except to tell you that what that revealed was that there was an increase of two particular chemokines that have been implicated in monocyte recruitment into tumors, CXCL13 and CXCL5, and a corresponding increase in CXCR5 and CXCR1 expression, the receptors, in the monocytes. And this increase happens specifically in the setting of cancer. So if we look just in mice that had MI but didn't have tumors, we didn't see the increase in CXCR5 or CXCR1 expression. So we knew that we had an increased supply of the Ly6E high monocytes. We're getting increased recruitment. And we wondered, once they're recruited, are their phenotypes different? So this is RNA-seq data on the myeloid, monocytic myeloid-derived suppressor cells. These are the ones that are Ly6E high that we isolated from the tumors. I believe this was at day 17. And you can see that there are significant changes in genes between the mice that experienced sham and the mice that experienced MI in the gene expression profiles of the mDSCs that we isolated from the tumors. We saw approximately 100 genes that were increased in their expression and 134 that were decreased in their expression. And what was interesting is when we looked at the genes that showed decreased expression in the mice that had experienced MI, the go terms that came up from this analysis were regulation of immune response, regulation of cell activation, lymphocyte activation, cytokine-mediated signaling pathways, interferon gamma signaling. So all of these responses that would be expected to restrict the tumor. In genes that were upregulated, it was more difficult to point to a specific function that might be happening to cause tumor growth, but the genes that were upregulated fell into categories such as regulation of system process, actin filament-based movement, intracellular signal transduction, and cell cycle process. Looking more closely at the genes that were downregulated that fell into those categories of being in the immune response, we saw genes whose expression are required for anti-tumorigenic processes, such as CD40, MHC class 2 associated genes, IRF4, which is important for mounting an anti-tumorigenic immune response. And by performing upstream regulator analysis, so looking at these genes that are changed and trying to predict what would regulate this upstream, we found that factors such as interferon gamma, STAT1, interferon alpha, so type 1 interferons, TLR3, were all inhibited, suggesting that, that these upstream regulators of these genes are being somehow suppressed in the tumor after MI, in the mDSC specifically. And in terms of ones that were activated, the major one that popped out was interleukin 10, which is, of course, an anti-inflammatory cytokine. So I mentioned that these mDSCs express Ly6E high and are monocyte-like, but in order to prove that they are, in fact, mDSC, you can perform a T-cell suppression assay. And this is an in vitro assay where you isolate the mDSCs from the tumors and co-incubate them with labeled CD8 positive T cells and look at proliferation and activation. And so we had two reasons for doing this. One, to prove that the cells that we had isolated and were doing the RNA-seq on met the definition of an mDSC, but also to see whether on a per cell basis, they were more immunosuppressive. Here in the white bars, these are the T cells alone. And you can see this is their level of interferon gamma and level of granzyme B, which is a marker of CD8 T cell activation. When we co-incubate with the mDSCs that we isolate from tumors of mice that had the sham surgery, you see that they can suppress interferon gamma expression. And when we then incubate with the mDSCs from the mice that experienced MI, you can see an even further suppression of interferon gamma and also a suppression of granzyme B upregulation, suggesting that that the mDSCs that we're isolating from the mice that had undergone MI are more immunosuppressive on a per cell basis. So we now know that there are more Ly6E high monocytes in the circulation, more getting recruited to the tumors. They have a different gene expression profile, and they're more immunosuppressive. To see whether they were actually required for our phenotype, we took advantage of this mouse. It's called the CCR2 depleter mouse. And CCR2 the diphtheria toxin receptor has been placed downstream of the CCR2 promoter. And basically, when you deliver diphtheria toxin, it allows you to selectively deplete CCR2 positive monocytes. And so this was our scheme. We used the EO771 implantable model and performed MI or sham surgery 3 days later, and then delivered the diphtheria toxin starting at day 10. Again, this was the time point where the tumors began to diverge, and followed the tumor growth out to 20 days. And these are our controls to show that we were effectively depleting the CCR2 positive monocytes. So this is in bone marrow. These are our sham mice. In the lighter color are the ones that are the depleter mice. And you can see that we've got a 60% decrease in the bone marrow in both the sham and MI, and a near depletion in the circulation in both sham and MI. And when we looked at tumor growth, we saw that depleting the CCR2 positive monocytes abrogated the phenotype that we saw. So in the red circles are mice, control mice that experienced an MI compared to sham. And you can see, again, we have this doubling in tumor growth. And when the CCR2 positive monocytes are depleted, this growth is ablated. And we don't see a significant difference between the sham CCR2 depleted and the MI CCR2 depleted. And we saw interesting changes within the tumors of these mice. We did not see a difference in the number of CD3 positive T cells, but when we looked at FOXP3 positive regulatory T cells, we saw that when we depleted the monocytes in the MI mice, we had a decrease in FOXP3. And this correlated with an increase in CD8 positive T cells in the tumors of the mice that had MI and the monocytes were depleted, and also an increase in granzyme B, the marker of CD8 T cell activation. And this suggested that these monocytes that were being recruited were both necessary for the enhanced growth phenotype, and also were driving this immunosuppression. So one thing that I found really fascinating was how these effects of MI on tumor growth were being sustained, because we're looking at tumor growth 20 days after the MI. So the life cycle of a monocyte in the circulation is just several days. It's not several weeks. And to me, this was reminiscent of what people had been talking about in terms of trained immunity. And we wondered whether MI could imprint an innate immune memory response. So whether MI as a sterile inflammatory event could epigenetically reprogram the bone marrow so that all of the monocytes that are being released 3 weeks later could still be changed from that initial event. And this is just a schematic to show the concept of innate immune memory. I think most of you are familiar, but basically there's an initial priming stimulus, whether it's BCG or beta glucan, and then a return to baseline. And upon a secondary stimulation, it can be the same stimulation or a different stimulation, you can see either a heightened response in the situation of training, or a blunted response in the situation of tolerance, such as LPS tolerance. And people that work in this field have shown that this reprogramming occurs through epigenetic changes in the chromatin, and also changes in cellular metabolism. So to begin to look to see whether MI was driving immunosuppression by epigenetic changes in the bone marrow, we performed a technique called ATAC sequencing. We isolated the bone marrow monocytes at, I believe this is 17 days post-MI, and And ATAC sequencing basically looks for these areas of accessible chromatin. So open areas where transcription factors and transcriptional machinery can access. So these are poised genes, genes that can be more readily transcribed versus ones that are closed or inaccessible. And when we compared the bone marrow monocytes from the mice experienced MI or sham, what we saw in blue was many more loci that were less accessible in the MI versus the sham. We also saw some that were more accessible. And we used an analysis tool called grade analysis to look at these loci and to look for what biological processes might be represented. And we saw in the ones that were more closed, so less accessible, regulation of the immune system, inflammatory response, leukocyte activation, cytokine production. So many of the same genes that we saw changed in the tumoral MDSCs were now less accessible in the bone marrow monocytes. And within these regions that are more closed, we tried to predict transcription factors, that transcription factor binding motifs that might be driving this response. And the ones that came out were ones like the pioneer factors, PU1 and CBP, as well as IRF8, which can work with PU1. And IRF8 is a very interesting one because there are studies that have shown that IRF8 downregulation or knockout in monocytes leads to an MDSC-like phenotype. So if all of these MDSC binding site and all these binding sites for IRF8 are less accessible, this could in fact potentially drive the MDSC-like phenotype. But to try to prove this a little, I should never use the word prove. My husband has a math degree, and anytime I say prove, he gets on my case. To demonstrate this further, this link further, we integrated the RNA-seq from the tumoral MDSCs with the ATAC-seq from the bone marrow Lys6E high monocytes and looked for genes that were both transcriptionally inhibited in the tumor and also had reduced chromatin accessibility in the bone marrow monocytes. And these are those transcription factors that we saw enriched. And among these genes are some really interesting ones in terms of an immune response, such as CD40, CD86. CLEC7 is dectin-1, which is the receptor for dectin-1, which is involved in innate immune training. But then the last piece of proof that we needed, not proof, the last piece of evidence that we needed to show that this was epigenetically imprinted was to show that it could be transferred to naive mice through a bone marrow transplant. And so we again use the CD45.1, CD45.2 congenic strains, and the E0771 tumor model induced the MI or sham. And at day 23, we harvested the bone marrow. We transplanted the bone marrow to naive mice and let them recover for 14 weeks. So over 3 months. And then we implanted the 80771 tumor and monitored growth for 26 days. And this is just showing that after 14 weeks of recovery, we had equal numbers of the transplanted cells. I'm blanking on the word, sorry. Showing that we had equal reconstitution of the bone marrow from the MI and sham group. And interestingly, the mice that experienced MI had a higher number of monocytes in their circulation than the ones that had bone marrow from the mice that had sham surgery. So we were transferring this monocytosis phenotype, and the tumors grew twice as fast. And to me, this was really striking because these are mice that have never experienced an MI, but they have bone marrow from mice that have that have experienced the MI, and that was sufficient to lay the ground for this increase in tumor growth. So we think that MI is accelerating breast cancer progression via long-term reprogramming of the myeloid compartment. There are many factors going on here. The MI is increasing hematopoiesis in the bone marrow. I didn't show you, but there are increased numbers of hematopoietic precursors. We have more Ly6E high monocytes in the circulation that have been already pre-programmed to be immunosuppressive before they are released from the bone marrow. There are more of them in the circulation, an increase in their recruitment to the tumor, and these monocytes then seem to set up an immunosuppressive environment where we have an increase in the mDSC content, an increase in the Treg cell content, and when we look at the phenotype of the mDSCs, they seem to have a more immunosuppressive profile. And so I think that this data is very compelling because I think, one, for breast cancer survivors, it's really important to understand this link between breast cancer and cardiovascular disease risk. We've known before that these patients are at increased risk for cardiovascular disease, but now we know that if they go on to have a heart attack, this could have a negative impact on their cancer and cause cancer recurrence and cancer-specific mortality. But also, I find it very interesting that MI can imprint this type of learned response on the hematopoietic system, and it opens the door to the possibility that other comorbid conditions will be affected. And that could be something like an infection, an acute infection, but also other comorbid conditions such as diabetes or other diseases where inflammation plays a major role. So I'll end by acknowledging all the people that did this work, particularly Graham Colwyn. He's now a postdoc at Memorial Sloan Kettering. Alexandra Newman, another postdoc who helped a lot with the T-cell suppression assays and some of the work needed in the revisions, as well as many other members of my lab that contributed. At NYU, our collaborators, Jonathan Newman, Ed Fisher, Tessa Barrett, and David Park, Lee Jones at Memorial Sloan Kettering, and Kaiser Permanente for their help looking at the epi data. And thank you all for listening. I'm happy to take any questions.
Thank you, Catherine. This was really a great opportunity for us to listen to this newly published paper from, the corresponding author. So thank you so much. Everybody can raise their hands from the participant window, just as a reminder, and I'll take the questions. And while you are, raising your hands, I'll ask my first question. So we did this in our journal club last week. So I have a question from
oh no,
from my group.
Great.
So Catherine, what happens to the heart in this scenario? Like these immunosuppressive myeloid cells, is that like something that the heart needs to recover from the ischemic reperfusion injury?
So we looked at this. We did two things. We did TTC staining just to look grossly at the remodeling in the heart. And then we also did echocardiograms on the mice. And David Park, who's listed on the acknowledgement slide, was the one that helped us. His group helped us with that. And so we were very grateful. And we did not see any differences in either the extent of damage to the heart or in the recovery. So these changes didn't seem to affect cardiac repair. It was more the long term. And I think in a way, it gets back to the idea of innate immune memory where you have an initial stimulus and then a second stimulus, right? And so the cancer is the second stimulus. whereas maybe the repair is still part of that initial wave.
Yeah, thank you so much. I think I see a hand already. I'll ask for Mabruka to just unmute herself. Thank you.
Thank you. That's really very great talk. I enjoy every single aspect of it. So I'm also interested in the heart tissue in terms of when you did the bone marrow transplantation of the MI mice into the healthy mice. Did you see any changes in the heart when you did this? And also, what could be the factor that's released from the heart after the insult and goes into the bone marrow to reprogram the immune system?
So your second question, that's the big question next, right? In the bone marrow transplant, we didn't actually look at the hearts. That's an interesting idea. We were focused more on the tumor growth and the monocytosis that we were seeing. And so that's something that we should go back and look at. In terms of what is the factor, so I think ischemia is potentially a major factor. And so we're going to try to address that by looking at different forms of ischemic injury. For example, if you were to ligate the femoral artery, do you see the same effect? If you ligate the renal artery, do you see the same effect? If you use inhibitors of hypoxia, for example, can you reverse it? But beta adrenergic stimulation also is known to have a major effect on the bone marrow. And we know that from Matthias Narendorf and Phil Schwarsky's work. And so that's another potential factor. And there was an interesting paper recently in Circulation where they used a pressure overload model in the heart, and they identified a secreted factor that they thought could accelerate cancer. So I think there are multiple possibilities, and we need to make our list and start knocking them down.
Thank you.
Thank you, Mabruka. So Zui, I'll ask you to unmute yourself.
Hello, our interesting study. So I'm just curious about the trained immunity, because you mentioned that metabolism also going to contribute this trained immunity. So I just wonder how the MI actually reprogram the bone marrow progenitor cell and also the, let's say, monocyte, and contribute to this phenotype.
Well, I wish I had an answer for you. We did the experiment. We tried to do metabolomics on the bone marrow monocytes. We didn't get an answer, mostly because it's very, very difficult to capture these cells and not have them change, right? To be able to do the metabolomics very quickly and not see a change. And so the experiment failed, and I don't have an answer for you. We tried valiantly a few times. And even though we did pilot studies and things worked well in our pilot study, on the day when we were harvesting, 8 mice per group, and there were more cells, more waiting time, then it just didn't work.
Oh, thank you.
But it would be very interesting to do it.
Okay. Yeah, another question is, based on what I'm reading from the innate, the trained immunity, some of this trained immunity actually, cause the pro-inflammatory monocyte. So, but your phenotype is, immunosuppressive phenotype. So I just wonder, what are your thoughts about the difference?
So training, people often use that term when they talk about a heightened response upon secondary stimulation. I sometimes use training, even though we're talking about a more of a suppressive response, only because I like the idea that we're training the immune system in a certain way. But people more often refer to what we're seeing as tolerizing. Right. So like the LPS tolerance effect that's been known for a really long time, where if you stimulate a monocyte with LPS and then come back a week later and re-stimulate, you have a much lowered cytokine response. So I think that that's what we're seeing. But even though initially we used models for training and tolerance that seemed very straightforward, it's a lot like the macrophage M1, M2, where we now know that in between M1 and M2, there's a big spectrum of, 1.1, 1.2, 1.3. And so I think that we'll see more of that in terms of innate immune training, because as people now look at different pathogens, different doses of the pathogen molecular pattern that are used, they're seeing different training responses. So I think there's going to be a wide spectrum there. It's useful as a model initially to define an in vitro, but then in reality, I think it makes sense that not everything is going to provoke the exact same response. There's going to be a spectrum.
Thank you.
Oh, sorry. I was speaking, but nobody was hearing me. Well, thank you again.
My pleasure. Nice to see everybody.
Nice to see you, and thanks for all the questions. So I'm going to hand it over to Lale because Rob is next, and Lale will be taking care of this. Thank you.
Oh, yes. Thank you, Catherine. It was a great talk. So next speaker is Rob Bauer. Rob was an assistant professor at Columbia University, Department of Medicine, and his lab focuses on the role of tribbles 1 in cardiometabolic disease, and also identification of novel targets in lipid droplet formation and vascular biology in atherosclerosis. So today he will talk about trouble with tribbles, functional validation of plasma lipid GWAS loci. Rob, if you can share your screen.
Great. Okay, can everyone see that and hear me?
Yes.
Yep, okay, great. So just first off, thank you so much to Lale and Ebru and Katie for not only inviting me to do this, but also for organizing this whole seminar series. I think it's been really great and a great way for the community to keep in touch throughout this weird time. So well done organizing this, and I hope it continues on in some form going forward. That was also a great talk from Catherine. Catherine's a tough act to follow on a normal day, and so when she's just published a Nature Medicine paper, it's even harder. So I'm going to do my best, but that was a fantastic talk. So wonderful. Thanks for sharing that. So I think this seminar series is a unique seminar series, and because of that, I'm going to give a little bit of an unorthodox talk, at least as far as I'm concerned today. Rather than go deep on one big story, I'm going to show like a couple little vignettes. And the reason I'm doing that is because the overall point I want to make today is about functional validation of GWAS loci. And in particular, that there is so much work to be done in that field. And I think there's a lot of consternation in the field, and perhaps rightfully so, about the fruits of that labor and the best way to do that and things like that. So I'm going to talk, I think, mainly about that today, but I'm going to use research in my lab to sort of illustrate my thoughts about that. So a little bit of a different talk than usual. But as I mentioned, my lab is broadly interested in functional validation of genome-wide association study loci, right? And this is the NHGRI GWA catalog as of actually a couple of days ago. And I actually I think I have to stop showing this figure soon because it's so overwhelmed with dots and associations at this point that it's hard to even make any sense of it, right? So as of Tuesday, there are 196,000 SNP trait associations across the genome from all the different GWASs that have been done. So literally almost the entire genome at this point is associated with some sort of trait or disease in humans, which is just an astounding amount of biology that has been unearthed by these genetic studies. Despite that massive amount of discovery, the functional validation has really lagged behind. And this is a paper that I really like, in part because they're saying all the things I believe, but this is from Mike Gallagher and Alice Chen-Plotkin at the University of Pennsylvania. And they wrote this a couple of years ago called The Post-GWAS Era from Association to Function. And I just have highlighted here this one line from the abstract that functional studies aimed at delineating causal genetic variants and biological mechanisms underlying the observed statistical association with disease risk have lagged, right? And they have a nice figure in this paper where they basically just counted up the number of GWAS studies that were being released every year And then they also tried to calculate the number of functional studies. That's obviously a little hard to do. They were sort of selective in how they figured that out. But I think the point remains that in 2016, where we're well into the GWAS era at this point, there were 3,800 GWAS studies published, and there were 84 functional follow-up studies published. So I really think that, if we aren't in the post-GWAS era at this point, we should be in the post-GWAS era at this point, because there is so much information out there that has just not been picked over yet, right? And there's a variety of reasons for that. So cardiometabolic traits are no different, right? There have been GWAS done for plasma lipid metabolism. Some of the most recent ones have looked at over 300,000 individuals looking for associations with total cholesterol, LDL, HDL, or triglycerides. By my count, there's a little over 300 genomic loci at this point that have been associated with plasma lipid traits, one or more plasma lipid traits in humans. And about 220 of them are being associated with lipid metabolism for the first time by these genetic studies, right? So that's many, many genes with no known role in lipid metabolism that have yet to be tackled, right? The same is true for coronary artery disease. So this is a really nice figure from a review that Say Katharison authored a couple of years ago in Nature Review Genetics, looking at what we know about CAD loci. So this is GWAS for coronary artery disease, independent of lipid metabolism. And the nice things here is that, number one, the genetics is highlighting pathways, right, that are relevant to coronary artery disease, like LDL cholesterol, triglyceride-rich lipoproteins, inflammation, and vascular remodeling, vascular tone. So, the genetics is highlighting these pathways as relevant to the disease, which is in and of itself useful information. But then there are also just the lists of genes again, right? So by my most recent count, there were 160 loci that have been identified for coronary artery disease, independent of any sort of lipid association. And those 160 loci are not all illustrated here, but you can see that even in this figure, which is from a few years ago, when they binned all these genes in different pathways, there was a variety of loci that were not in any pathway, that it was just unclear what exactly the role was here. So the point of these opening slides is there's just a tremendous amount of biology still out there that has been highlighted by these GWAS studies that no one really knows what it's doing. And it's really just an amazing trove of potential discoveries. So my lab and myself for years have been tackling this issue of GWAS follow-up by mainly focusing on one locus, on chromosome AQ24. And this is a locus that has been associated with all 4 plasma lipid traits, as well as coronary artery disease. And these associations have been validated over and over again. So the fact that it hit these 5 associations has raised the interest in this locus for quite a long time. And if you go in and look at the actual GWAS signal, you can see here all the significant SNPs and the different lead SNPs. And these SNPs lie around 50 kilobases downstream from the gene TRIB1. So there's really not much else in this region. There actually, I don't have it on this figure, I realized a little bit ago, but there's actually a link RNA present here as well that has had some interest in it also. But the initial observation of this GWAS locus, given that TRIB1 was really the only gene nearby, led people to speculate that that's the gene of interest at this locus, right? And so we've been studying TRIB1 and the mechanisms underlying this genome-wide association for years now. So what is TRIB1? TRIB1 is a pseudokinase. There's 3 family members in humans, TRIB1 through 3. And by pseudokinase, what that means is that these proteins actually have very high homology to serine threonine kinases, but they're missing key amino acids in the catalytic domain, so they're not able to phosphorylate anything. Instead, they act as signal transduction modulators or scaffolding proteins. They promote the docking and interaction of other proteins by binding them and bringing them together. There are a variety of different functions and mechanisms that have been elucidated across the family, but the main function that's been investigated for TRIB1 is that TRIB1 can bind E3 ligases to promote the ubiquitination of target proteins. And in particular, for TRIB1, the best studied target is the transcription factor CEBP alpha. So TRIB can bind CEBP alpha, bring the E3 ligase COP1 into proximity of CEBP alpha, promote its ubiquitination and subsequent degradation. And at the time of its discovery, given its the strong associations with plasma lipoprotein metabolism, there were initial studies done in the liver, both by myself and other people working with Dan Rader, showing that if you overexpress TRIB1 in the livers of mice by AAV, you see a dose-dependent reduction in cholesterol and triglycerides, plasma cholesterol and triglycerides. And then in the liver-specific knockout, you see the opposite reciprocal phenotype. You see an increase in total cholesterol, non-HDL cholesterol, and triglycerides in the plasma, suggesting that TRIB1 in the liver, hepatic TRIB1, is important for lipid metabolism. And in fact, its activity decreases plasma lipids in mice. Through my work on that mechanism, what we were able to show is that a lot of what TRIB1 does in the liver is mediated by this protein CEBP alpha. There is a inverse relationship between these two, which I'll talk about in a minute. But we have approached going forward with this research, and in particular focusing on this locus, on looking at both TRIB1 and CEBP alpha and the roles that that regulatory axis is playing in lipid metabolism. In the liver, we found that CEBP alpha was regulating de novo lipogenesis and triglyceride production, and that might be feeding into regulation of lipidation of ApoB and VLDL secretion. So going back to this main point of functional validation of GWAS loci, in my opinion, there's three main difficulties in doing this, three main questions that have to be tackled anytime you're looking at one of these GWAS loci. The first is, what is the gene being identified at the locus, right? So if there are numerous genes at a locus, that can make the selection of the gene very difficult, right? And that's not even getting into the idea that maybe it's not even a gene, maybe it's a link RNA or something else, right? The second is, what is the function of the gene product, right? So a lot of these genes have no previously known role in lipid metabolism. So whatever the mechanistic link is completely unclear. Some of these genes have proteins that are completely uncharacterized and no one knows what they do. So there's a lot of work to do in that area. And oftentimes knowing the function is the only way to know the direction of effect of the association. So a GWAS doesn't necessarily tell you whether gain or loss of function is going to be beneficial or detrimental. All you know is that there's an association with that thing at that locus to the phenotype you're interested in. And then finally is, what is the tissue of interest for that gene that you've identified? So again, GWASs don't necessarily tell you what tissue you're supposed to be in, since DNA is the same across all different tissues, right? So you have to do the actual investigations to see which tissues are the one of interest. And what we found is that there can be some really interesting differences across different tissues. So I'm going to just walk through these three questions and show a little bit of data from my lab that shows how we're kind of going about tackling these things. So starting with, what is the gene being identified at the locus? Now, without getting too didactic here, one of the main observations of the GWAS era is that GWAS SNPs are predominantly non-coding. So they lie in non-coding regions around nearby genes. Sometimes they're surrounded by clusters of genes, and it can be very challenging to infer which gene is the gene of interest, because as we know now, the genome is very three-dimensional. And you might have a lead SNP here that's closest to gene A, but in reality, there's folding over of the genome, and this enhancer that this SNP lies in is interacting with gene C, which is downstream, right? So it's not as easy as it used to be. Historically, the closest gene was the choice, and we know now that it's far more complicated than that. So one of the things that we've been trying to do in my lab is use functional genomics to confirm the genes of interest at different GWAS loci. And to give an example, there's this locus that's come out of plasmolipid GWAS studies on chromosome 19 that associates with triglycerides and HDL in humans. And this signal has been assigned to this gene PEPD, peptidase D. And the reason for that is because if you look at the GWAS signal, the lead SNP here, this RS731839, falls in an intron of this gene peptidase D. So that's why that was assigned as the gene, because it falls in the intron of that gene. But because of our interests, one of the reasons that this became apparent to us is because right downstream here is CEBP alpha. And we've always been interested in the fact that CEBP alpha doesn't appear to be a GWAS target for plasmolipid metabolism, even though our functional data in mice suggest that it is a key regulator of lipid metabolism. So this raised the hypothesis that perhaps this SNP is not recognizing PEPD, but rather recognizing CEBP alpha. And in support of that hypothesis, if you go into ENCODE data and look at enhancer marks in hepatoma cell lines, so this is primary hepatocytes and HepG2 cells shown in pink here, and looking at DNA, open chromatin, and H3K4 monomethylation and acetylation, what we see is that here's that SNP, that's the GWAS SNP, that there's clearly a hepatocyte-specific enhancer that is present right around the region of the SNP, supporting the idea that this element here is doing something in hepatocytes. So we set out to use functional genomics approaches to try to prove that perhaps this is regulating CEBP alpha. We initially used CRISPR gene editing to cut out this region surrounding the SNP here. So we designed two guides to target this region, did the knockouts, did clonal expansion and genotyping of individual clones to pick colonies, got some wild type ones, some heterozygous samples you can see here, and then some full knockouts where we completely deleted that region. For those of you that are doing gene editing and screening clones like this, just for the record, it took us 90 colonies, screening 90 colonies to get 3 wild types, 9 hets, and 3 knockouts. which we thought was pretty good actually. And then we just did gene expression for CEBP alpha in those cells, and you can see the results here. And sure enough, when we looked at the heterozygous deletions, we saw a significant reduction in CEBP alpha gene expression. And when we looked at the homozygous deletions, we saw an even more significant reduction in gene expression. So confirming that that region does play a role in regulating CEBP alpha expression. Now, to my further surprise, we actually saw no significant changes in CEBP gamma or PEP-D, which are the genes nearby. Now, you know, I don't know that I necessarily would put a ton of stock in the idea that there's no effect there. My initial thinking was that it was probably going to affect all the genes, and you can see that this is pretty noisy in these regions. So, so it could be with further analysis, you might see something there, but the data is the data. We didn't see any significant changes there, while as we did with CEBP alpha. To try to go at this a different way, we also did transcriptional repression via CRISPR-I. This is, for those that are not familiar, this is a catalytically dead Cas9 that's bringing a transcriptional repressor CRAB domain into contact with the region of the genome that you assign your guide towards, basically rendering it transcriptionally inactive in a 1 to 2 kb window around your guide RNA. We designed 4 different guides, one that landed right on the SNP, one that sort of was directly in the middle of that big enhancer that I showed, one that targeted the transcriptional start site of CEBP alpha as a positive control, and then a non-targeting negative control. And we did this in HUH7 cells that were stably expressing dCas9. And sure enough, what we saw was basically the same thing, that if you target this enhancer element or the SNP, you do see significant reductions in CEBP alpha gene expression, and we don't see the same thing happening with CEBP gamma and PEP-D. So this pretty much conclusively shows that at least in HUH7 cells, that enhancer region is regulating CEBP alpha gene expression. And it may be that in the GWAS literature, this locus is being assigned to the wrong gene, that this is actually capturing a regulatory element that targets CEBP alpha downstream. So some ongoing work in the lab related to this functional genomic stuff. We're trying to do functional assessment of enhancer gene interaction through CRISPR-based approaches like I just showed you, but we're doing both candidate genes that we're just interested in for physiology, as well as GWAS loci. What we'd like to do, we're doing it mostly individually, but we're trying to scale up to do parallel, with the idea being that actually given single cell RNA-seq technology and things like that, we should be able to do functional assessment of pretty much all the enhancers at all the different hepatic GWAS loci, at least as far as we can infer at the same time. So that's something that we're trying to move towards. We're also trying to invest a lot in CRISPR dropout screens to identify relevant GWAS genes. I think this is one of the real powerful pathways forward for the GWAS community, is doing these CRISPR knockout screens for relevant phenotypes to see which genes at GWAS loci are popping up in those screens, because that shows actual functional relevance. We have an intense focus on the TRIB1 GWAS locus. We're doing a lot of functional work there, and then we're still following up on the role of CEBP alpha in plasma lipid regulation. So the second question I mentioned, is what is the function of the gene product, right? That's oftentimes the only way to know the direction of effect. And as I laid out to you before, we had established this mechanism in hepatocytes in mice that TRIB1 was downregulating CEBP alpha, and CEBP alpha normally upregulates de novo lipogenesis. But the nature of this interaction was really unclear. It was not clear from any previous studies what exactly the role was of TRIB1 in regulating CEBP alpha in hepatocytes. So we've done some actually very basic, just in vitro biochemistry work with some overexpression models to try to pick this apart a little bit and learn more about what TRIB1 in hepatocytes is actually doing. So we've made HUH7 cells that stably overexpress doxycycline-inducible GFP or TRIB1, and we can get really nice overexpression of TRIB1 in these cells. And one of the things we looked at was, did this mimic the phenotype, the opposite phenotype that we saw in the mice? So in our mice, when you knock out TRIB1, you get big increase in CEBP alpha. In HUH7 cells, so human hepatoma cells, we see that if you overexpress TRIB1, you get a nice reduction in CEBP alpha. So we were able to mimic that phenotype in vitro. The next question we asked was, is this a proteasomally dependent process, right? So that's the basic understanding of how TRIB1 works. So we use the MG132 proteasome inhibitor in cells overexpressing TRIB1. So these are all cells getting doxycycline, everything's overexpressing. And then this is without or with MG132. And we saw that that was able to rescue CEBP alpha expression in these cells. So this is a proteasomally dependent process. And then we took it a step further and knocked down COP1, the E3 ligase COP1, to see if that would also rescue CEBP alpha protein levels, and it did. So we've effectively shown that this model that has never been tested in hepatocytes is actually true in human hepatocytes or hepatoma cells, that TRIB1 is downregulating CEBP alpha in a COP1 and proteasome dependent manner, suggesting that that is the mechanism at play. But we've of course been wondering what other interacting partners are involved in this. There's so little known about TRIB1, and it seems unlikely that the only thing it's interacting with is COP1 and CEBP alpha. So we've done some standard tandem affinity purification approaches where we've overexpressed TRIB1 with a 3-by-flag HA tag, done pull downs with both the different tags, and then done proteomics to identify all the binding partners and compared those findings to the same experiment done with a GFP HA 3-by-flag tag. And what we see, this is the results of those proteomics, is so on the x-axis here, I have the fold change over GFP, and on the y-axis, I have the negative log p-value or FDR, and I'm only showing things that were enriched in the TRIB1 region, right? Because I wouldn't be interested in things that are enriched in the GFP pull down here, right? And so basically that means that the genes up here are kind of the most interesting ones. When we zoom in here, and I'm sorry, I know the resolution on this is not that great, but when we zoom in here, what we see is probably the most significant hit we get is TRIB1, which is good. That was what we would expect. We also identify MALT1. So MALT1 is a component of the NF-kappa B signaling cascade. And Warren Pear, who's interested, University of Pennsylvania, who's interested in TRIB1 and leukocytes, has done a little bit of work on TRIB1 and MALT1. So we were happy to see that as a positive control. But then the other two proteins that have popped out are these proteins STIC40 and STIC38. Now these were very interesting to us because these two proteins are themselves both pseudokinases. And in particular, there's actually a lot of similarities between STIC40 and TRIB1. So STIC40, first off, has very high protein homology to TRIB1. The sequences are highly homologous between the two of them. They have a lot of the same protein domains. Whole body knockout of STIC40 is perinatal lethal. That's the same as what we've seen with TRIB1. When you knock out TRIB1, those mice are also perinatal lethal. The expression patterns in humans closely mimic TRIB1. So wherever TRIB1 is highly expressed, STIC40 is almost always highly expressed as well. And then last is that previously Steve Blacklow up at Harvard was able to show that that STIC40 actually binds to COP1, the same E3 ligase that TRIB1 seems to be working through. So this set up the nice paradigm that STIC40 might be somehow interacting with TRIB1 and COP1 and regulating that function. We decided to test that by doing an siRNA knockdown of STIC40 in our overexpression cell lines. And sure enough, just like with treatment with the MG132 or with treatment with the siRNAs towards COP1, we were able to see that there was a rescue of the downregulation of CEBP alpha when we knocked down STIC40. So this data suggests that STIC40 is required for TRIB1 degradation of CEBP alpha, although the nature of that requirement is completely unknown right now. So there's a lot of, again, going back to the larger theme here of GWAS follow-up, there's actually a lot of very basic biology that needs to be done with a lot of these genes, right? A lot of these known mechanisms have not been validated in relevant cell types. They've not been validated in human cell types, and they've not been further explored at all to see what are maybe some of the other interacting factors at play. So we've been trying to go really deep on describing the TRIB1 functional mechanism in hepatocytes, as well as some other cell types that we're interested in. And the ongoing work with respect to some of the stuff I just showed you, I'm not showing you everything I have here just because of time restraints, but the nature of the interaction with STIC40, is it a direct interaction or an indirect interaction via COP1? Validating that interaction in vivo, we're seeing this in cells now, but can we show that this is true in vivo in mice and livers or in other tissues? Looking at a ubiquitination screen in HRA7 cells with TRIB1 overexpression to see what other targets are potentially being ubiquitinated. And then finally, we have a large body of work going on looking at TRIB1 turnover in hepatocytes. So, the therapeutic model here would be that increased TRIB1 levels would be good. And we've actually seen in vitro that TRIB1 protein is very rapidly turned over. So we're wondering if there's a way to effectively target that. So that's some ongoing work there. And then finally, the last question I think is really important when it comes to these GWAS loci is, what is the tissue of interest, right? So as I mentioned, GWAS does not inform tissue relevance. All of the TRIB1 work that I've shown you till now has mostly been in livers and in hepatocytes, and that's because of the strong interactions with plasma lipoprotein metabolism, the GWAS interactions, as well as there's also a GWAS association with circulating ALTs. We believe that's due to the association between TRIB and CEBP alpha and de novo lipogenesis. So ALTs are a marker oftentimes of liver health. So a lot of that early focus started in the liver, but there's actually another GWAS association with the same SNPs in this region between TRIB and plasma adiponectin. Now, adiponectin is a hormone that is involved in insulin sensitization, as well as some known roles in regulating plasma lipids in coronary artery disease. but of main focus, it is exclusively secreted from adipocytes. So if TRIB1 is playing a role in regulating adiponectin secretion, then that must suggest that TRIB1 actually has a function in adipocytes. Here's a look at the GWAS signal. So again, I'm taking you to the same plot, looking at the triglyceride associations. This adiponectin SNP is right in that exact same signal here. So further highlighting the fact that this is the same region that we're looking at, and suggesting perhaps that maybe even these SNPs are having an effect on adipose TRIB1. So maybe it's adipose TRIB1 that's actually driving all of these different associations. That's a possibility, right? Or it could easily be multiple tissues. So to try to answer that question, we set out to make an adipose-specific knockout mouse. We took our TRIB1 CKO mouse and crossed it to the adipoQ mouse to get efficient deletion of TRIB1 in adipocytes. And one of the first things we did was we looked at plasma adiponectin levels, as that was the association that got us here. Sure enough, we saw that in both males and females, just chow-fed TRIB1 ASKO adipose-specific knockout mice, they have increased circulating adiponectin, and that does not come with a change in mRNA levels. So we don't think this is due to a change in the transcription of adiponectin at all. Much to our surprise, these mice also had a plasma lipid phenotype. So they actually have reduced, this is just chow-fed TRIB1 ASKO mice, they have reduced total cholesterol, HDL cholesterol, non-HDL cholesterol, and triglycerides. And just to remind you, that's actually the opposite direction of the phenotype we saw in the liver-specific knockout. So liver-specific knockout of TRIB1 in mice increases plasma lipids. Adipocyte-specific knockout of TRIB1 in mice decreases plasma lipids. So really underscoring the point that looking at different tissues for these GWAS loci is incredibly important, because the direction of effect can be completely opposite. And one of the questions in the lab is, which one of these tissues is dominant in mice, but also both in humans, right? That's an ongoing complicated question. So we've been trying to do a little bit of work trying to understand both what is the function of TRIB1 in adipocytes and how is it regulating adiponectin secretion, as well as plasma lipid metabolism. For the adiponectin phenotype that we saw, we collected SVF from the mice and differentiated them to adipocytes and just measured adiponectin in the media over the cells. And sure enough, we find increased adiponectin from the ASKO adipocytes compared to the wild type ones, suggesting that this is an adiponectin secretion phenotype. When we actually look inside the cells too, we see that the cells themselves have more adiponectin compared to the wild type mice. And we still do not see any change in mRNA levels, right? So, I mentioned before that we've been looking at this from a TRIB1 CEBP alpha centric viewpoint. CEBP alpha is a well-known regulator of adipogenesis and the adipogenic transcriptome. We do not believe that CEBP alpha is driving this phenotype because we don't see any changes in the transcription of TRIB1. That would leave the idea that TRIB1 is somehow regulating the proteasomal degradation of adiponectin. So we've taken this same SVF and treated them with MG132 to see if that abrogates the phenotype, brings the wild type mice back up to the knockout mice. And what it turns out is we do not see that. So when you treat SVF with MG132, one interesting thing you see is that that actually drastically downregulates adiponectin transcription to equal extent in both cell types. We don't see any relative change in adiponectin between the groups, right? So if we look in the conditioned media, that magnitude of difference remains the same. Nothing changes with the MG132. And then when we look in the cells, we see that there's no change when we treat with MG132. So we don't think that adiponectin is being turned over by the proteasome. We don't think that that is contributing to this phenotype. We also don't think it's CEBP alpha because we don't see any transcriptional changes. So what that means is not only do we have a different direction of effect in adipocytes, but we actually have a completely different mechanism at play in adipocytes. Again, further underscoring the need to move into different tissues. So we've been trying to understand that mechanism. We've been using some of the same unbiased proteomics approaches. I mentioned the TAP pull down. In this instance too, one of the things we've done is turbo ID proximity biotinylation. So we fused TRIB1 to a promiscuous biotin ligase and transfected it into 3T3L1 cells and pulled down proteins that were biotinylated, which presumably are in close contact to TRIB1. And we've done the proteomics on these guys, found the proteins that were either exclusively found in the TRIB1 data sets or drastically upregulated in the TRIB1 data sets in both the TAP pull down as well as the turbo ID. Have intersected those data sets to see which genes we're finding in common between these two approaches, and then have done David pathway analysis to look at what these proteins are. And the main molecular function that we're identifying in these 13 genes is poly-A RNA binding. and positive regulation of translation. So it appears that TRIB may be actually regulating translation in these cells, and that might be the link between TRIB and adiponectin. And then, of course, the last question is, what's the relationship to the plasma lipids? And we have a lot of ongoing work with respect to this, but just one finding that we found that's pretty interesting is we've done RNA-seq on the adipocytes from the TRIB1 ASKO mice versus wild-type mice. We find an upregulation in LPL transcription and a downregulation in ANGPTL4 transcription. That would suggest an increase in LPL activity because ANGPTL4 is a negative regulator of LPL. We've actually also done secretomics from adipose tissue from TRIB1 wild type and ASKO mice, and we see trends that agree with that RNA-seq. We see increased LPL secreted into media from ASKO cells, and we see decreased ANGPTL4 secreted into media. So this is all consistent with an increase in LPL activity. And then sure enough, we've done fluorescent lipid substrate cleavage assays that we learned from Ira Goldberg's lab down at NYU to look at LPL, adipose-specific LPL activity in the ASKO mice, and we see that those mice have an increase in LPL activity. And we think that that probably underlies the triglyceride phenotype, and we're curious if that might be part of the link to the cholesterol phenotype that we see. So some ongoing work related to the adipocyte TRIB1 work is validation of interaction with those RNA binding proteins. That's a big focus for us, and maybe even doing some more global translation approaches, look like polysome profiling or RiboSeq to find other targets that may be differentially translated. Looking at the connection between TRIB1 and ANGPTL4 and LPL, we don't actually know what the direct link between those two is, so we're trying to figure that out. We have a couple ideas. And then, of course, the role of adipocyte TRIB1 in cholesterol metabolism. One would think the LPL phenotype would be more of a triglyceride phenotype, but there's ways it could be related. And so I'll just close, with thanking all the people that did this work, but really also just trying to sum this up that, to just reiterate my initial point. We've had it's been very exciting and interesting to study this TRIB1 locus. There are so many different directions to take it in because it's such an open-ended question. There's so little known about this gene that we've been able to really chase this down in a lot of different directions. And there are literally hundreds of other loci out there from these GWAS studies that have the same potential to be, unearth all sorts of different novel biology. And I again have sensed some consternation in the field that maybe GWAS isn't paying off that much, and is it really worth to do this follow-up? And I think it is worth doing it. It's obviously hard, and it's hard to get funding for it, but I do think it is a worthwhile endeavor for the field. And I hope that some of the data I've shown you today supports that idea. So just real fast, I'll thank Elizabeth Ha, who's a really talented graduate student in my lab who's driven forward all the adipocyte-specific knockout work. Caio Mateus is a technician in my lab who's actually been doing all the in vitro HUH7 cell work. Alan and Krista are PhD students in my lab who are also helping out with a lot of the functional genomics and getting some of our CRISPR screens off the ground. And Noel Walsh and Kavita Jadhav were ex-members of my lab who actually did the specific functional genomics that I showed you earlier today. And thanks again so much for the invite to talk today. I really appreciate it, and I'm happy to take any questions anyone might have.
Okay, excellent, Rob. Thank you. I know Elizabeth's amazing work actually, part of the third part of it, but it was really nice that you gave us like an overall general, look into the GWAS and how it differentially regulates different tissues. So I will start again, everyone, if you have a question, please use the raise your hand function. I see that Ebru has a question, but let me start first. There's clearly association of TRIB1 with adiponectin, and that raises the question about its role in adipocytes. But I'm wondering whether you see a change in adiponectin levels in the liver TRIB1 knockouts. Could it be possible that liver somehow talks to adipose tissue for adiponectin, secretional levels?
Yeah, so that's a great question, and I just didn't have time to show the data. When we did our RNA-seq experiment, we looked at actually, do I have it right here? We looked at here we go. We looked at adipocytes. We isolated adipocytes from the ASKO mice, and then we also isolated liver from the same mice. And we did RNA-seq in both tissues from the same mice. And what we found is that there was, depending on how stringent you want to be, if we go with the more stringent, FDR 0.05 and a twofold change, there were 2,600 differentially expressed genes in the adipocytes and there were only 19 differentially expressed genes in the liver. So in my mind, this sort of supports an idea that it's the adipose that's driving this and not the liver. That being said, we have not forgotten about the liver. We have done individual like qPCRs to look at downstream adiponectin receptor genes to see if those are differentially expressed. We haven't really seen that in the chow-fed mice. We've also done a little bit of physiology. I mean, we've looked at like VLDL secretion and things like that just to see if there's anything going on. We don't have any evidence supporting a role for the liver right now, but I have not ruled it out for sure.
All right, sounds good. I think Ebru has a question.
Hi, Rob. So maybe this is too preliminary to talk about, but what do you think are the translational targets of triples? It's really
well,
interesting.
that's a great, yeah, I mean, that's a great question. You know, I don't know, right? So, I mean, one of them is adiponectin, right? That's clearly one. We know that there's more adiponectin. We don't think it's being turned over faster. We don't think it's being transcribed more. I mean, if you just think about like the steps that a protein goes through, right? That doesn't leave too many other options, right? Other than just the translation of the protein from the RNA. You know, I didn't get into this because I have the slide here too. I didn't plant these questions, but I did bring all these slides. I didn't get into this, but the CEBP alpha story is kind of confusing here as well. So when we look at CEBP alpha in these mice, we actually do see increased CEBP alpha protein levels. You can see that right here. But we don't see any increase in CEBP alpha message levels. And we also don't see any difference with respect to CEBP alpha protein turnover. So it appears that TRIB is still regulating CEBP alpha levels, but it doesn't appear to be doing that by the same mechanism it did it in hepatocytes. So we wonder if perhaps CEBP alpha is also a translational target of TRIB1. And then that, of course, feeds into a whole like adipogenesis story, but we don't really see any evidence that there's any changes in adipogenesis. So it's complicated. But so to specifically answer your question, what are the translational targets? The only two I would say as of right now are adiponectin and CEBP alpha. Those are the two proteins we know there's increased protein levels. They're not increased transcription, and they're not being differentially proteasomally degraded. But I'm sure there's others.
Yeah, very interesting. Thank you, Rob.
I have one more short question. So you showed data with the STIC40, right? That was really interesting. How about STIC38? Are they related, 38 and 40? And yeah.
Yeah, so I have not forgotten about STIC38. You know, I think the observation that STIC40 binds COP1 combined with the fact that we just can't do everything at once is why we sort of started at STIC40. One of the only other mechanisms that has been described about TRIB1 is its potential role in regulating MAP kinase signaling. And there's been some in vitro data shown in the past that TRIB can regulate MEK-ERK signaling and ERK phosphorylation. So it turns out that STIC38, probably the thing it's most studied about is MAP kinase signaling. And I wonder if perhaps TRIB and the interaction between TRIB1 and STIC38 is the branch off to MAP kinase signaling. So that's something that we're interested in. These proteins are all definitely fairly homologous. You know, there's actually a really nice review from Patrick Ayres, who's a gentleman that studies pseudokinases in the UK. And if you branch off the phylogeny of all the kinases and pseudokinases in the human genome, these are all definitely a family unto themselves, these STIC proteins and the tribbles proteins. So they have a lot of homology, but the interactions have just not been explored. And to my knowledge, there's no studies looking at STIC40 and STIC38 in hepatocytes for the most part.
Oh,
and I will note that STIC40 is expressed in adipocytes also. So
that's exciting. More work, I guess, for the future. Okay, all right. If no one has a question, we'll thank Catherine and Rob again for 2 amazing talks, and we'll continue next week. See
Thanks,
you,
everyone.
Thank you, Rob, and bye, everyone. Bye.